Welcome to the Deep Dive. Our aim, as always, is to cut through the noise and give you the essential insights. Today, we're digging into the research on a really foundational molecule in immunology.
That's right. We're talking about Thymosin Alpha-1, or TEN1 for short.
Exactly, Thymosin. It comes up a lot in peptide research.
It absolutely does. You could almost call it a cornerstone.
Yeah.
It's maybe not the loudest signal in the immune system, but it functions like a very precise molecular instructor.
A molecular instructor, I like that.
Yeah.
So our goal today is to synthesize what we found in the research, stack the history, how it actually works, the latest applications.
And really pull out the important nuances, right? Not just the headlines. We've got documents covering immune modulation, research limitations, all of it.
Okay, let's get into it. This stuff is fascinating, the biochemistry, potential uses, but, the sources themselves start with a really critical warning. It's in those documents about the purpose and limitations of research peptides.
Yes, absolutely crucial. We have to address that right up front.
We do. So what's the key message there?
Well, it's crystal clear in the literature. When we discuss these powerful peptides like Thymosin, which are derived from natural sources, they are strictly, and I mean strictly for research purposes only.
Okay.
Strictly research. The explicit directive, and we need to be impartial reporters here, is that these compounds are not intended for human consumption, not for self-administration. We're discussing the published science, operating entirely within the regulatory framework for lab use.
Got it. So that boundary is set. Everything we talk about today relates to scientific literature on compounds designated purely for research.
Precisely.
Right then. With that necessary framework in place, let's dive into the science itself because it really is remarkable. Okay, so where does Taiwan actually come from? You said it's naturally derived, so it's not something cooked up in a lab from scratch?
No, not originally. It's something our bodies make. The story really kicks off back in the 1970s, which was a really pivotal time for immunology as we know it.
Right.
Taiwan was first isolated, identified properly back in 1977, and where they found it is super significant, the thymus gland.
The thymus. Okay, most people might vaguely remember that from biology class. It's important early on, right?
Hugely important, especially in youth. It's essentially the primary school, or maybe the boot camp, for our immune system's T lymphocytes, those critical adaptive immune cells.
Ah, okay. So the T cells learn their job in the thymus. It makes sense then that a key peptide from there would act as an instructor.
Exactly. The name fits perfectly.
So structurally, what is this instructor? You mentioned it's a peptide, which means it's made of amino acids.
That's right. And it's actually quite small. T1 is just a short chain, only 28 amino acids long. But what's really interesting is how it's made in the body. It's not built directly. It's actually cleaved or cut from a much larger precursor molecule.
The precursor.
Yes, a protein called prothymosin alpha. The fact that it needs to be processed like this tells you it's part of a carefully controlled biological pathway. It's not just some random fragment floating around.
Only 28 amino acids. Seems small, but you hinted earlier it's quite potent. Are there specific chemical properties that make it stable or effective?
Definitely. Its molecular weight is around 3,108 daltons, which is pretty small for a biological molecule. But two features really stand out for its stability and function. First, its N-terminus, the starting end of the peptide chain, is acetylated.
Acetylated? What does that do?
It's a common chemical modification, like capping the end. It basically protects the peptide from being chewed up too quickly by enzymes in the body, gives it a longer shelf life, so to speak, so it can deliver its message.
Okay, protection. And the second feature.
Second, it's a highly acidic peptide, and it's known to be quite heat stable. And when it's in solution, like in our bodies, it tends to fold into a specific shape, predominantly an alpha helical conformation.
An alpha helix.
Right.
Like a corkscrew shape. Why is that shape important for its job?
That structure is vital. It contributes significantly to its stability, as we mentioned, but it's also likely crucial for how it docks with its specific receptors on immune cells. It presents the right key to fit the lock, if you will. It's a robust structure, which helps explain why it works reliably under different biological conditions.
Makes sense. So going back to the molecular instructor idea, what's the main lesson TEL1 is teaching the immune system? Is it just telling the body, make more soldiers?
Not quite like that. It's more subtle. It doesn't really trigger the bone marrow to churn out brand new immune cells. Its role is more about refining the existing troops.
Refining them.
Yeah, it helps educate and activate the cells that are already there, especially those T lymphocytes we mentioned, the highly specific fighters of our adaptive immune system. TEL1 essentially helps them perform better, making the response more effective and more targeted.
So it's not about quantity, but quality control. making the existing immune force smarter, better coordinated, making sure they react appropriately.
That's a great way to put it, smarter and more coordinated. As one of the research summaries pointed out, Taiwan really acts as a bridge. It connects, classic T-cell immunology with the modern understanding of cell signaling pathways, and even touches on regenerative research. Just by influencing those fundamental communication lines between cells, it's all about improving the conversation within the immune system.
Okay, if it's improving the conversation, we need to know who it's talking to first. How does TAMA1 actually interact with immune cells? The sources point very clearly towards toll-like receptors, the TLRs.
Yes, TLRs are absolutely central here. Think of TLRs as the immune system's frontline surveillance system. They're part of the innate immune system, the first responders. Their job is to recognize broad patterns associated with danger.
Patterns like parts of bacteria or viruses.
Exactly. Specific, highly conserved molecular structures, bits of bacterial walls, viral RNA or DNA. These receptors are constantly scanning for these non-self patterns.
Right, but TAMA1 it's a human peptide, so how does it end up triggering receptors designed to detect foreign invaders? That seems a bit odd.
It is fascinating, isn't it? It suggests a really deep level of integration in how our immune system is regulated. TAO1 acts as what's called an endogenous ligand, a signal originating from within the body, but one that can interact with these pattern recognition pathways.
So it's like an internal signal that uses the same alert systems.
Sort of, yes. Or perhaps it modulates how that system responds. The research shows it interacts with multiple TLRs, not just one. Specifically, the papers mention TLR2, TLR4, but also TLR3, TLR7, and TLR9.
Okay, why that particular combination? Does interacting with TLR3, 7, and 9 tell us something specific?
It does, because those three, TLR3, 7, and 9, are primarily located inside the cell in compartments called endosomes, and they specialize in detecting nucleic acids.
Nucleic acids.
Right.
Like DNA and RNA.
Precisely. TLR3 recognizes double-stranded RNA, often a sign of viral replication. TLR7 picks up single-stranded RNA, common in many viruses. And TLR9 detects specific DNA sequences, unmethylated CpG DNA, found in bacteria and some viruses.
So by engaging these specific TLRs, TAO1 is effectively priming the cell to respond to threats that are often intracellular, like viruses hiding inside our cells.
That seems to be a key part of its function, yes. It sets the stage for a particular type of immune response, one geared towards intracellular pathogens.
Okay, so the TLR is triggered. That's step one. What happens next inside the cell? The sources mention these downstream signaling pathways, NF-key, STAT, and IRF3.
Yeah.
Can you unpack those for us? They sound important but complex.
They are absolutely critical controllers. Think of them as relay stations that carry the signal from the receptor on the cell surface or inside the endosome down to the cell's nucleus where the genes are.
Okay.
So when TAO1 triggers the TLRs, these pathways get activated. Let's take NFA first, nuclear factor kappa B. This one is like a master switch for inflammation and immunity.
The master switch.
Pretty much. When NFB gets activated, it moves into the nucleus and turns on hundreds of genes for inflammatory molecules, for cell survival, for mobilizing other immune cells. If you need a strong immune reaction, you generally need NFP involved.
Okay, that's NFP. What about STAT and IRF3?
STAT pathways, signal transducer and activator of transcription, are often activated by cytokines, those immune messenger molecules. They play roles in cell growth, differentiation, survival. They relay specific messages. And IRF3, interferon regulatory factor 3, this one is really key for antiviral responses.
Antiviral.
Yes. Activating IRF3 leads directly to the production of type I interferons. Interferons are, well, the body's primary defense mechanism against viruses. They interfere with viral replication and alert neighboring cells.
So Taiwan isn't just sounding a general alarm via NFP. It's also specifically activating the antiviral machinery through IRF3.
That appears to be the case, yes. It's orchestrating a specific kind of response.
Which brings us to the outputs. What actual molecules, what cytokines get produced as a result of all this signaling?
The research highlights a really important trio: interleukin-2, IL-2, interleukin-12, IL-12, and interferon gamma, IFN-OA. These are powerful communication signals for the adaptive immune system.
Okay, IL-2, IL-12, IFN gamma. What do they each do?
Well, IL-12 is crucial for pushing T cells towards becoming cytotoxic T cells, the killers that target infected cells. It promotes what we call a Th1 response. Interferon gamma, IFNR, is a major player in that Th1 response too. It activates macrophages, makes cells better at killing pathogens, really ramps up cellular defenses.
And IL-2.
IL-2 is essentially a main growth factor for T cells. Once a T cell is activated, IL-2 tells it to proliferate, to make copies of itself, building up an army of specific responders.
Okay, so Thymosin is boosting some powerful pro-inflammatory signals like IL-12 and interferon gamma. This seems like it could be risky. How does the research explain this without causing, say, a cytokine storm or just general overactivation? You mentioned modulation earlier.
Yes, and that distinction is absolutely critical, especially when you think about using something like this therapeutically. Even in research, just blindly boosting the immune system isn't always good. It can lead to autoimmunity or excessive inflammation.
Right.
Thymosin is described as a modulator or a balancer. The idea is that it strengthens the necessary defense, particularly that Th1 type cellular response we mentioned.
The one good against viruses and cancer cells.
Exactly. But it does so while helping to maintain regulatory control. It's not just pressing the accelerator, it's also ensuring the brakes are functional. It aims for a balanced, effective response, not just a stronger one.
So tuning the system, not just turning up the volume. Tell us more about this Th1 polarization you mentioned. Why is leaning towards Th1 important?
So broadly speaking, adaptive immune responses can be categorized. Th1 responses are primarily cellular, involving cytotoxic T cells, activated macrophages, really good at dealing with threats inside cells like viruses or cancer. Th2 responses are more antibody-focused, often involved in fighting parasites or in allergic reactions. T1 seems to preferentially promote that Th1 profile. It nudges the immune system towards the tools best suited for intracellular battles, making the response more efficient and appropriate for those kinds of threats. It helps focus the immune effort.
That kind of focused effort requires really good communication between all the different immune cells. How does TUTA1 help with that internal chatter?
It significantly enhances the role of certain key communicators, especially dendritic cells.
Dendritic cells. They're the ones that present antigens, right?
Yeah.
Show the T cells what the IMU looks like.
Precisely. They're often called the professional antigen-presenting cells, or maybe the intelligence officers of the immune system. TUTA1 promotes their maturation.
What does maturation mean for a dendritic cell?
A mature dendritic cell is much better equipped to do its job. Specifically, maturation increases the expression of important molecules on its surface, things like co-stimulatory molecules, which give the T cell a crucial go signal, and MHC class I molecules.
MHC class I. Remind us why that's so important.
MHC class I is like the display window on almost all our cells. Cells use it to show little fragments, peptides of whatever proteins they're making inside. If a cell is infected with a virus, it'll display viral peptides on MHC class I. If it's becoming cancerous, it might display abnormal cancer peptides.
So the T cells can see what's happening inside the cell.
Exactly. Cytotoxic T cells patrol and inspect these MHC class I displays. TUTA1, by boosting MHC class I expression on dendritic cells and potentially other cells, makes antigen presentation more efficient. It ensures the threat alert is displayed clearly and effectively to the T cells.
Better presentation, clearer alert. Makes sense. What about the D cells themselves, the ones receiving the message? Does TUTA1 affect them directly?
It does. The research shows it influences T cell responsiveness directly. One key mechanism is upregulating the high-affinity IL-2 receptor on the T cells.
The receptor for that IL-2 growth signal we talked about?
Yes. Remember, IL-2 tells T cells to multiply. By making the T cells express more of the high-affinity version of the IL-2 receptor, TUTA1 essentially makes them much more sensitive to that growth signal.
So it's like giving the T cells better antennas to pick up the multiply now command.
That's a perfect analogy, better antennas. So when the T cell recognizes its target antigen and gets the co-stimulation, it can respond much more robustly to the IL-2 signal, leading to rapid proliferation and a stronger overall counterattack. It optimizes the whole activation loop.
Okay, this picture of TUTA1 as an optimizer, improving recognition, communication, and response. It immediately makes you think about situations where the immune system isn't working optimally, like with aging.
Absolutely. This is where TUTA1 research becomes incredibly relevant from a broader health perspective. That thymus gland where TUTA1 originates, it doesn't stay highly active forever.
It shrinks, doesn't it? Thymic involution.
Exactly. After puberty, the thymus gradually shrinks and becomes less active. This means fewer new T cells are produced and educated, and, logically, the levels of thymic peptides circulating in the body, including type 1, also tend to decline with age.
And what are the practical consequences of that decline in TUTA1 and thymic function?
The research consistently connects these lower levels with observable weaknesses in the immune system of older adults. We see generally weaker immune responses, reduced effectiveness when they get vaccines.
Like the flu shot.
Yes, exactly. Reduced vaccine responsiveness is a big one. And also slower recovery times if they do get infections. The immune system just isn't as robust or as quick to adapt.
So the idea in the research is that maybe Thymosin could be used to sort of prop up that declining thymic signaling, to try and counteract some of that age-related immune weakness.
That's precisely the line of investigation. Can we support the natural signaling environment even as the gland itself is less active to maintain better immune function? And the vaccine data is particularly compelling here.
What does it show?
Studies have found that older individuals who happen to have higher circulating levels of these thymic peptides naturally tend to mount much stronger and, importantly, more durable antibody responses after getting vaccinated, say against influenza.
More durable meaning the protection lasts longer.
Yes. It suggests that supporting this take-up pathway might be key not just for the initial immune response, but for building lasting immune memory, which often wanes with age.
That potential naturally leads into major research areas. Let's start with chronic viral infections. Where has Thymosin been studied there?
There's quite a long history here. Research has focused heavily on viruses like hepatitis B, HBV, hepatitis C, HCV, and also HIV. These are all chronic infections where the virus manages to persist, often because it finds ways to suppress or exhaust the T-cell response over time.
Right, the immune system kind of gives up or gets worn down.
Exactly. So in research models related to these viruses, Thymosin has been investigated for its ability to sort of reinvigorate that response. The idea is to promote that Th1 type T-cell activity we talked about, restore a better cytokine balance, and help the immune system regain control over the virus.
And what about more recently with the COVID-19 pandemic? Was Thymosin looked at there?
It was, yes, quite rapidly, actually. Given its known immunomodulatory effects, it was evaluated as a potential supportive agent, especially in patients with severe COVID-19.
Why severe cases specifically?
One of the dangerous features of severe COVID was often lymphopenia, a really sharp drop in the number of lymphocytes, including T cells. The immune system was getting depleted. Some research reports suggested that TO1 might help reduce this lymphopenia and improve overall immune homeostasis, basically,
Yeah.
help stabilize the immune system during that critical phase.
Interesting. Helping restore balance during severe stress. That concept of restoring competence seems directly applicable to oncology research, too. The sources say Thymosin is being looked at as an immunotherapy adjunct. What does that mean?
An adjunct means it's used alongside other treatments, not necessarily as a standalone therapy. In cancer, this is really important because the patient's immune system is often compromised.
Compromised by the cancer itself or the treatments?
Both, actually. Many cancers actively release signals to suppress the immune system, to hide from it. And then, traditional treatments like chemotherapy and radiotherapy, while targeting the cancer, are often broadly immunosuppressive as a side effect.
So the immune system is fighting a war on two fronts.
Precisely. And this is where adding something like TG1 in research settings becomes interesting. The hypothesis is that it can help restore some of that lost immune competence.
How? By boosting T-cell function again.
Yes, potentially by enhancing T-cell recognition of tumor antigens, improving dendritic cell function as we discussed, and helping T-cells proliferate even in that challenging environment. Studies are looking at combining Thymosin with standard chemoradio and also with newer approaches like immune checkpoint inhibitors.
Checkpoint inhibitors being the drugs that take the brakes off the immune system.
Exactly. The idea is Thymosin might help tune up the T cells so that when the brakes are released by the checkpoint inhibitor, the response against the tumor is even stronger and more effective.
Are there specific examples in the source materials where this combination showed promise in research?
Yes. One example highlighted was in research models looking at hepatocellular carcinoma, which is a particularly tough type of liver cancer.
Right.
In some of these studies, combination protocols that included Thymosin showed longer progression-free survival compared to the standard therapies used alone. The suggested mechanism ties right back to enhancing antigen presentation and restoring T-cell responsiveness, allowing the immune system to better target the cancer.
So it's potentially helping the immune system see the target better and respond more vigorously, even when other factors are trying to suppress it.
That's the rationale being explored. It's about amplifying the body's own defense mechanisms in a targeted way.
Okay, one more area mentioned is severe immune dysregulation, specifically sepsis. This sounds incredibly challenging.
It is. Sepsis is a life-threatening condition. It's basically the body's extreme, dysregulated response to an infection. You get this chaotic mix of widespread inflammation. The cytokine storm idea can be relevant here, but also, paradoxically, profound immune suppression often develops too. The system just crashes.
A real balancing act gone wrong. So Thymosin, being a modulator, was thought to potentially help restore some equilibrium.
That was the hypothesis, yes. Given its ability to promote Th1 responses but also support regulation, researchers thought it might help stabilize patients caught in that dangerous storm and crash cycle.
And what did the research find? Did it work?
This is where we need to be really clear about the nuance. The results have been mixed. Some smaller, often preclinical studies or early-phase clinical research did suggest some promising immune supportive effects in sepsis models.
But
But when researchers tried to confirm these findings in larger, more definitive clinical trials, the results often didn't show a clear, consistent benefit across the board.
why the difficulty in translating those early results? Sepsis seems like exactly the place you'd want immune balance.
It's incredibly complex. Sepsis isn't one single thing. It's a syndrome with many different underlying causes and trajectories. Patients are highly diverse. Their baseline immune status, the type of infection, when in the sepsis cascade they are treated, it all matters hugely.
So applying a single modulator like 1001 uniformly might not work for everyone.
That seems to be the challenge. Some patients might be in a hyperinflammatory phase where modulation could help, while others might already be deeply immunosuppressed, needing a different kind of intervention. It highlights that while TANO1 is a sophisticated tool for rebalancing, the sheer complexity and rapid changes in severe sepsis likely require even more personalized, stage-specific strategies. It's not a magic bullet for that specific complex condition based on the larger trials.
That sepsis example really underscores the complexity we're dealing with. But let's pivot to safety. For any compound, especially one modulating the immune system, safety is paramount for continued research. What do the sources say about Taiwan's track record in research settings?
This is actually one of the strongest points in Taiwan's profile, and a major reason why research has continued for so long and across so many areas.
Why is it considered so safe?
Well, fundamentally, because it's so similar to a peptide our bodies naturally produce. It's not a completely foreign chemical. And across decades of preclinical work and numerous clinical studies for various research purposes, it has consistently shown a really excellent safety profile.
Good tolerability.
Very good tolerability, yes. It's been observed to be well tolerated in a wide range of research populations: healthy volunteers, people with chronic infections, cancer patients undergoing treatment, so including immunocompromised individuals.
Were there any side effects noted at all, even minor ones, in these research studies?
The side effects reported are generally minimal. The most common thing noted is usually just mild, temporary reactions right at the injection site, a bit of redness or discomfort, that sort of thing.
Nothing systemic or long-term?
Crucially, no. The sources emphasize that longer-term studies haven't revealed any significant signs of toxicity. No major issues with blood counts, no organ damage, no evidence of it building up in the body in a harmful way.
That's exactly what you'd hope for from something designed to work with the body's natural systems.
Absolutely. Its strong safety record, combined with its biological origins, really makes Tenwen one of the best characterized and most reliable immunoregulatory peptides available for research today. It provides a solid foundation to build upon.
Okay, so building on that foundation, where's the research heading now? What are the exciting new frontiers for Taiwan beyond the established areas like chronic viruses and oncology support?
One really promising area that's gaining a lot of traction is using Taiwan as a vaccine adjuvant.
An adjuvant, something you add to a vaccine to make it work better.
Precisely. Given everything we've discussed about how TELA1 enhances T-cell responsiveness, dendritic cell function, and the cytokine production, all key elements of generating immunity, researchers are actively exploring if adding TELA1 to existing or new vaccines can boost their effectiveness.
How would that work?
Yeah.
By making the initial immune response stronger.
Yes, and potentially longer lasting, too.
Yeah.
There have been research studies, for example, with vaccines against influenza and hepatitis B, where adding TELA1 to the vaccine formulation led to higher levels of protective antibodies being produced.
Higher antibody titers, right?
Exactly. And perhaps even more importantly, some studies suggest it leads to more durable immune memory, that longer lasting protection we talked about earlier in the context of aging. So it could be a way to improve how well vaccines work, especially in populations who don't always respond optimally, like older adults.
That makes perfect sense. And what about the combination studies in cancer research? Are they getting more sophisticated?
Definitely. The research isn't just looking at TAO1 with traditional chemo anymore. It's moving towards highly specific combinations. Pairing TAO1 with targeted therapies that hit specific cancer mutations, or, as we touched on, with those immune checkpoint inhibitors.
Trying to create synergy.
Exactly. The goal is to use TAO1 to kind of prime the immune system, maybe overcome resistance mechanisms the tumor is using, and ensure that these powerful cutting-edge treatments can do their job most effectively. It's about optimizing the entire anti-tumor immune environment.
Fine-tuning the attack. Okay, finally, the sources mentioned innovative delivery methods. If the peptide works, how are researchers trying to get it to the right place more effectively?
Right, because how you deliver a biological molecule can drastically affect how well it works. Systemic injection isn't always the most targeted approach. So there are a couple of exciting frontiers here.
What's the first one?
Nanoparticle-based delivery. This involves encapsulating TAO1 inside tiny engineered nanoparticles.
What's the advantage of that?
Several potential advantages. The nanoparticles can protect the peptide from degrading too quickly in the bloodstream, potentially allowing for lower doses or less frequent administration. And depending on how they're designed, these nanoparticles could even be targeted.
Targeted to specific cells or tissues.
Potentially, yes. You might be able to design them to be preferentially taken up by immune cells or to accumulate in specific tissues like lymph nodes or even tumors, concentrating the effect where it's needed most.
Very clever. And the second innovative approach mentioned?
This one is really fascinating. Probiotic delivery?
Probiotic delivery. You mean using beneficial gut bacteria to deliver TAO1?
That's the concept being explored, yes. The idea is to genetically engineer harmless beneficial bacteria probiotics so that they actually produce TAO1 or perhaps carry it as cargo.
And deliver it where? Directly into the gut.
Exactly. Deliver it right into the gut environment. We know the gut microbiome and the gut's associated lymphoid tissue, the GALT, play a massive role in regulating our overall immune system.
The gut immune axis.
Precisely. So, by delivering TAO1 directly to this critical interface using probiotics, researchers hope they might be able to modulate the immune system in a very localized, perhaps more natural way, leveraging those gut immune connections. It opens up completely new research avenues. #OutrodOutro.
Wow. Okay, this has been an incredibly detailed look at Thymosin Alpha-1. We've really unpacked its significance. Clearly a cornerstone in immunological peptide research.
Absolutely. We've seen its role in educating T cells, fine-tuning cytokine signals.
And really striving for that crucial immune balance.
And that balance is the key theme, I think. TAO1 isn't about brute force. It's subtle, it's regulatory. It fits perfectly with this idea of working with biological systems, optimizing them rather than just overriding them, leveraging the body's own logic.
Right. And that brings us to our final thought, something for you, the listener, to consider. Given everything we've discussed, the natural decline of the thymus with age, the research showing TAO1's potential in enhancing vaccine responses, in supporting cancer immunotherapy.
Yeah, where does that lead us?
Well, what if future research increasingly focuses not just on treating sickness after it happens, but on proactively supporting these native messenger systems like TAO1 throughout life? Could we potentially maintain a more robust, resilient immune architecture as we age?
Moving beyond just reaction towards optimizing the underlying foundation of immunity across the lifespan. That's a really powerful concept to think about.
It is. And just to circle back one last time, for absolute clarity,
Mhm.
everything we've discussed today is drawn strictly from published scientific research literature.
And the compounds themselves, including TAO1, are designated for research purposes only. They are not for human consumption or self-use. That boundary remains firm.
Thank you for joining us on this deep dive. We hope this exploration sparks your curiosity.